Hello everyone, my name is Sneha. I am a student of BSc Chemistry Honours 6th semester. Today I will make a video for visually impaired students on the topic fermentation from organic chemistry. Fermentation is the chemical transformation of organic substances into simpler compounds by the action of enzymes, complex organic catalysts which are produced by microorganisms such as molds, yeasts or bacteria. Fermentation technology is the use of microorganisms to produce food, pharmaceuticals and alcoholic beverages on a large-scale industrial basis. The science of fermentation is called zymology. Principle of fermentation process. The basic principle involved in the industrial fermentation technology is that organisms are grown under suitable conditions by providing raw materials meeting all the necessary requirements such as carbon, nitrogen, salts, trace elements and vitamins. The end products formed as a result of their metabolism during their lifespan are released into the media, which are extracted for use by human beings and that have a high commercial value. Types based on respiration. Aerobic fermentation. Aerobic fermentation or aerobic glycolysis is a metabolic process by which cells metabolize sugars via fermentation in the presence of oxygen and occurs through the repression of normal respiratory metabolism. Aerobic fermentation means that oxygen is present. Wine, beer and acetic acid vinegar such as apple cider vinegar need oxygen in the primary or first stage of fermentation. When creating acetic vinegar, for example, exposing the surface of the vinegar to as much oxygen as possible creates a healthy flavorable vinegar with the correct pH. Lactic acid fermentation. Lactic acid is formed from pyruvate produced in glycolysis. NAD+ is generated from NADH. Enzyme lactate dehydrogenase catalyzes this reaction. Lactobacillus bacteria prepare curd from milk by the type of fermentation. Also, during intense exercise, when oxygen supply is inadequate, muscle derive energy by producing lactic acid. Alcoholic fermentation. This is used in the industrial production of wine, beer, biofuel, etc. The end product is alcohol and CO2. Pyruvic acid breaks down into acetaldehyde and CO2 is released. In the next step, ethanol is formed from acetaldehyde. NAD+ is also formed from NADH, which is released in glycolysis. Vacant and some bacteria carry out this type of fermentation. Enzyme pyruvic and decarboxylase and alcohol dehydrogenase catalyze these reactions. Citric acid. Citric acid derives its name from Latin word citrus with a molecular weight of 210.14 gram per mole, which contains the carboxylic functional group with three different values of pKa 3.1, 4.7 and 6.4. Citric acid, a primary metabolic product, is the most important organic acid produced in large quantity and is extensively used in food and pharmaceutical industries. Citric acid is a tricarboxylic acid formed in the tricarboxylic acid cycle or Krebs cycle and is formed in small quantities in all living organisms. Citric acid was first commercially produced in England in 1826 from lemons until 1919. It is produced mainly by submerged fermentation using Aspergillus niger or Candida from different sources of carbohydrates such as molasses, and starch-based media. Citric acid production. About 99% of world citric acid production occurs through microbial processes which can be carried out using submerged or surface cultures. The 70% of the anhydrous or monohydrate form of citric acid is used in food and beverage industry as an acidifier or antioxidant to preserve or enhance the flavors or aromas of fruit juices, ice cream, and marmalades. 20% is used in the pharmaceutical industry as antioxidant to preserve vitamins, effervescent, pH corrector, blood preservative, or in the form of iron citrate as a source of iron for the body, as well as in tablets, ointments, and cosmetic preparations. In the chemical industry, which uses the remaining 10%, is employed as a foaming agent for the softening and treating of textiles. Surface fermentation using rice bran or other starch or sugar rich substrates have been used for surface fermentation by Aspergillus niger or in liquid solution in flat aluminium or stainless steel pans. A niger can be used to produce citric acid despite the high content of trace metals in rice bran are used. The citric acid is extracted from the bran by leaching and is then precipitated from the resulting solution as calcium citrate. Submerged fermentation. It is a common practice where citric acid is made. The fermenter is made of acid resistant materials such as stainless steel. The carbohydrate sources are molasses decationized by ion exchange, sucrose or glucose. MgSO4.7H2O and KH2PO4 at about 1% and 0.05 to 2% respectively are added. The pH is never allowed higher than 3.5. Copper is used at up to 500 ppm as an antagonist of the enzyme aconitase, which requires iron. 1.5% of methanol, isopropanol or ethanol when added to fermentation containing unpurified materials increase the yield. Copper is used at up to 500 ppm as an antagonist of the enzyme aconitase, which requires iron. 1.5% of methanol, isopropanol or ethanol when added to fermentation containing unpurified materials increase the yield. The yields are reduced in media with purified materials, as high aeration is deleterious to citric acid production. Mechanical agitation is not necessary and air may be bubbled through antifoam